Okay, class, last section we talked about transcription and what that was, and that was making RNA from a DNA template. This section we are going to look at translation and what translation is, and after this lecture you should be able to summarize translation, and you should be able to explain the relationship between genes and proteins. Okay, so what is translation? Translation is actually the decoding of mRNA message into a polypeptide chain. So you're actually changing messenger RNA into the polypeptide protein. It takes place on ribosomes. So actually mRNA is transcribed in the nucleus. And we already found that out from transcription, okay? And mRNA enters the cytoplasm and it attaches to the ribosome. And then tRNA comes in, and that carries only one type of amino acid. So we have the tRNA here, and that actually attaches to the mRNA using the anticodons. And then, as I just told you, that tRNA already has anticodons. These anticodons are complementary to mRNA. So just like the DNA replication, where the base pairs were A and T and then C and G and vice versa, those ones, we found out that in RNA, instead of T's, we have U's, so it's A and U and C and G. Anticodons for tRNA are the same as for messenger RNA, so A's and U's will go together and C's and G's will go together. As each codon of a mRNA molecule moves through the ribosome, the proper amino acid is brought into the ribosome by the tRNA. So it brings in whatever amino acid that needs to come in, and it links up. You can see the linkage right here, that the tRNA links to the mRNA, and then it breaks off. So this messenger RNA right here, with the anticodon of UUU, attaches to the codon of AAA, brings in lysosine, so then that lysosine can attach to whatever amino acid this is. So the amino acid is then transferred to the growing polypeptide chain, and you'll see this peptide chain right here start to continue to grow, and we'll have a link of all the polypeptides. The ribosome forms a polypeptide bond between the first and second amino acid. So you can see that bond forming right here. At the same time, the ribosome breaks the bond that is held for the first tRNA molecule and its amino acid, and then releases the tRNA. So it brings in that tRNA, it attaches to the mRNA, and then that amino acid, it attaches to the amino acid chain, which is the polypeptide chain, and then it breaks off, so then the tRNA can go get another amino acid. The ribosome moves to the next codon, and the tRNA molecule brings the amino acids specific to the mRNA codon. So what I just said keeps happening. The polypeptide chain continues to grow until the ribosome reaches a stop codon on the mRNA molecule, and we'll go ahead and talk about that further. So it keeps going until the mRNA sequence reaches a stop. The ribosome then releases a newly formed polypeptide, which would be the protein, so it releases this polypeptide, and then it releases the mRNA. So let's review again. So we have DNA at the beginning, and that in transcription is then made into RNA, mostly messenger RNA. And then through translation, which uses tRNA, it breaks down into the proteins. It makes proteins. So now you can see how DNA is all related to the proteins and vice versa. Without DNA, we cannot have the proteins. So let's start off with a strand of DNA. And I want to put this all together for you in the huge concept that you should see it in. So we have DNA strand A here. And we are going to go ahead and, go ahead and get my pen out. And we are going to replicate this DNA. So we're going to make a new DNA strand B. And so we're going to replicate. replication. Maybe I should switch to white so that you can see it. Ink color, white. Okay, so during that replication, we actually are going to do the base pairing with that. So if it's a T, it has to be an A. If it's an A, it has to be a T. If it's a C, it's going to be a G. And these are just the base pairs. So it's a G. And so you just go through and you're doing the base pairs. And this is just for the replication. So if I ask you to do a replication, you would know how to do it. So G, T, A, T, T, A, T, G, 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 oh, no, it's going to be C, sorry, excuse me, C, A, C, and then that is going to be T, A, G, tag. Okay, so the next would be the transcription. Transcription. I-O-N. Transcription. Okay. So transcription, we are making messenger RNA. And to make the messenger RNA, you have to use the first strand of DNA, and it's going to be very, very similar to the second strand, the B strand of DNA, but the only difference is that all the T's are going to be changed to U's because we have uracil instead of thymine. So it's going to be A-U-G, C-G-C, U-U-U, C, or G, U, A, U, U, A, U, G, G, C, A, C, then U, A, G. So that would be the messenger RNA for this first strand. Okay, next we have translation. And translation, we need to go ahead and make the tRNA. The tRNA goes off of the mRNA. And so if we have an A, then we have a U, and then we have an A, because we have a U there, and then we have a C, and then it would be G, C, G, and so this would be very similar to the first strand of DNA, except for there is a U there. U's take place of the T's in the first strand. So then we would have next one would be A, and then C, A, U, A, U, and then A, C, C, G, U, G, and U, oh, no, it'd be A, U, G, AUC, AUC. Oh, I messed up big one on that one. Last one's AUC. Sorry, that's my fault. Okay, so I know it's kind of confusing. So just remember that if you're doing DNA, okay, it's going to be A and T, C and G. Okay, if you're doing RNA, anything with RNA, whether it be tRNA or mRNA, okay, you're going to have A and U and then C and G. G and C. And those ones are always going to be together. So if there's a G here, there has to be a C here. If there's an A here, there has to be a U. And that's how you how you do the transcription of them. So this is the first step.
Bauer's going home. Have a good night.
Bye, Mr. Bauer. I just did a shout out to you in my video.
See you tomorrow.
Yeah. Shout out to Mr. Bauer. So this is the first step of transcription. The second, or translation, this second step of translation is actually naming these three things. Putting a name to these three codons. Okay? And actually we name the messenger RNA codons. So we're going to name these codons, okay? And we are going to do that on the next slide, I believe. Yep. So AUG, remember I told you before that we were going to use this to name it. So I have this sequence for the messenger RNA that was on the previous slide. So if you go back, I have the messenger RNA and I just have them listed for you. So we're going to practice naming here together. So if you want to move faster than me, fine, then you can check your answers, that's fine. So to first start off, you're going to start with the yellow. And we're going to start here. So we have A, then we're going to go to the little bit darker yellow, which is U, and then we're going to go to G, right there. So we're going to have methionine, which is a start codon. And you know methionine is a start codon because it's the only one. Okay, it's the one that normally everything will start with. Then we have a CGC. So let's go to C, then go to the yellow C, and then or G, and then go to the darker yellow C, which gives us the next amino acid, which is arginine. That's an A. Sorry, my pen's not working. Next one, U. So we have U, phenylalanine. Then G, U, A, valine. Then I have U, A, leucine. Then I have U, G, tryptophan. And these are all amino acids. And then linked together, those amino acids are going to build your polypeptide chain. And then we have C, A, C, histidine. Oh. Should be a T. And then next we have U, A, G, which is the stop codon. So it means stop. So then that's where that polypeptide chain would stop. They wouldn't go any further. It's going to end there. So that's how you would name it, and that's when I was talking about the stop codon. So once it reaches that stop point, it's just done. It's not going to go any further. So hopefully you don't have any questions. Hopefully I cleared up some of the unclear things for you. I will have this chart right here for you when you do your worksheet. Your worksheet, you will have to do transcription and translation and name. So that's why I wanted to work with that for you. If you have any questions, make sure you ask me so then I can clear up anything that I need to clear up for you. Have a great day.